Hello dear students, welcome to Bio Prime Points. In this video, we are going to discuss some very essential points from the chapter Biotechnology Principles and Process of the second session. Okay, the first prime point, what are the major processes of recombinant DNA technology? So here, the first process is isolation of DNA. Isolation of DNA from a donor cell. Then, after isolation, the DNA is broken by using restriction endonuclease. That process is called restriction digestion. That is the fragmentation of DNA by using restriction endonuclease. Then, the third point is the isolation of desired DNA fragments. From that isolated DNA, we require only a particular fragment. So that isolation of that desired DNA fragment is possible by a mechanism. That mechanism is called gel electrophoresis. Then the isolated DNA fragment is amplified. The amplification of gene of interest by using PCR. PCR means polymerase chain reaction. Amplification means production of large numbers of copies. Then after amplification, the ligation of the DNA fragment into a vector using DNA ligase. That means the isolated DNA fragment is attached to a vector DNA. So when two such DNA fragments are integrated, we can create a recombinant DNA. The ligation means joining. Joining of the DNA fragment into a vector DNA using an enzyme called a DNA ligase. That is how we are creating a recombinant DNA. Then the next process is the transfer of recombinant DNA into a host organism. The mechanism is called host transformation. Then once it is transferred into a host organism, the host organism is cultured. The culturing of the host cells on a suitable medium in a large scale. We are using bioreactor. That is the culturing of host organism. When the host organism is cultured, the DNA will be functioning and producing the desired product. Then the extraction of the desired product thus created is called recombinant protein. And the final mechanism is the downstream processing. That means the product is produced, the recombinant protein is produced, then it must be made ready for marketing. That is the final process. That is the final finishing of the product. That process is called downstream processing. So in a particular recombinant DNA, the important processes are isolation of DNA, fragmentation of the DNA by restriction digestion, isolation of the desired fragment using gel electrophoresis, then amplification of the gene of interest by using polymerase chain reaction, the ligation of the DNA fragment to the vector DNA by using for the creation of recombinant DNA, then transfer of the recombinant DNA into a host organism. The mechanism is called host transformation. Then the culturing of transformed host in using bioreactor for producing recombinant protein on large scale. Then finally, the finishing of the final finishing of the desired protein, the recombinant protein by a mechanism. That mechanism is called downstream processing. Okay, so these are the important protocol of biotechnology or recombinant DNA technology. Okay. The next prime point is gel electrophoresis. Gel electrophoresis is for isolation of desired fragment. The fragments of DNA are separated by a technique called gel electrophoresis. Agarose, a natural polymer obtained from the seaweeds, is used as a medium matrix. The DNA fragments being negatively charged are separated by forcing them to move through a matrix towards anode under an electric field. The DNA fragments separate or resolve according to their size. That means the smaller fragments will move faster and the larger fragment will move in a slower manner. The separated molecules are stained by using a stain. That stain is called ethidium bromide and visualized by exposure to UV radiation. Then these separated fragments will be seen as bright orange colored bands. The separated bands of the DNA on the gel are cut out from the gel by a process. That process is called elution. Anyway, these mechanisms are collectively for isolating a desired fragment of DNA, and the mechanism is called gel electrophoresis. And the enzymes used in the DNA extraction for cell digestion, we are using lysozyme enzyme for extracting DNA from bacterial cells, chitinase enzyme for extracting DNA from fungal cells, cellulase enzyme for extracting DNA from plant cells. For protein removal, proteases are used. Then, for removal of RNA fragments, ribonucleases are used. Then, for precipitation of purified DNA, chilled ethanol is used. Okay, so this is for isolation of DNA from a desired cell. If it is a bacterial cell, we are using lysozyme. If it is a fungal cell, we are using chitinase. If it is a plant cell, we are using cellulase. Then the product obtained, the DNA obtained may be having some proteins, integrated proteins and RNA that can be removed. The proteins are removed by using protease enzyme. RNA fragments can be removed by using RNase enzymes or ribonuclease enzymes. Then the purified DNA can be extracted in the form of using chilled ethanol. Okay. The next very important bioprime point is the polymerase chain reaction, PCR. This is for amplification of the gene of interest. The desired DNA can be made into large numbers of copies by using this mechanism called polymerase chain reaction. PCR is a machine-mediated process by which the DNA fragments can be amplified into desired number of times. This technique involves three main stages. First one, denaturation. Second one, primer annealing. Third one, extension or polymerization. These are the three mechanisms. What is meant by denaturation? Denaturation means the double-stranded DNA is broken down into two single-stranded fragments. And this is made possible by heating up to 95 degrees centigrade so that the hydrogen bonds between the two strands will be broken and the dsDNA is separated as ssDNA fragments. Okay. The denaturation refers to the conversion of one double-stranded DNA into two single-stranded fragments by breaking the hydrogen bonds in between them. This is achieved by heating up to 95 degrees centigrade. Okay. Next one, primer annealing. Primer annealing refers to the addition of two primers at the 3' end of the separated DNA fragments. This occurs at 55 degrees centigrade. The primers are chemically synthesized short segment of RNA or oligonucleotides that are complementary to the segment of DNA. The primers are attached or added to the 3' end of the separated DNA fragments. Then, extension or polymerization refers to the synthesis of DNA strand based on the template DNA strands by adding corresponding DNA nucleotides to the RNA primer in 5' 3' direction. This occurs at 75 degrees centigrade. Based on attached to the RNA primer, new complementary DNA segments are added, nucleotides are added so that a new DNA, a complementary DNA will be produced. This is achieved by using a thermostable DNA polymerase enzyme, Taq polymerase. Because the process takes place at 75 degrees centigrade, a thermostable DNA polymerase is needed. The thermostable DNA polymerase is obtained from Thermus aquaticus archibacterium, a thermophilic archibacterium called Thermus aquaticus. That enzyme is called Taq polymerase. Okay, it's a very important point. Polymerase chain reaction. Three stages are there: denaturation, annealing, extension, or polymerization. Okay. Next bioprime point is host transformation. What is host transformation? The entry of the recombinant DNA into a host organism. The recombinant DNA is entered into a host organism so that the host organism gets transformed. The bacterial cell must be made competent to take up the DNA by treating them with a specific concentration of calcium that increases the efficiency with which the DNA enters the cell through the cell wall pores. This is called electroporation. That means the foreign DNA fragment can be entered into a cell. The cell is called host cell. So for that, the host cell must be prepared. The host cell must be made competent. That is by a process called electroporation. That means treating the cell in a divalent cation like calcium, the pore size will be increased. Then the recombinant DNA can be forced into such cells by incubating them with the recombinant DNA on ice, followed by placing them at 42 degrees centigrade. At first 0, then 42, again then putting back down ice. This is what is called heat shock. So by electroporation and heat shock, a cell can be made competent, a host cell can be made competent and ready for absorbing the recombinant DNA. Okay. Then there is another method in the case of animal cells, a method called microinjection, in which the recombinant DNA is directly injected to the nucleus of the animal cell by using the microneedle or micropipette. It's a direct injection of recombinant DNA into the animal cell nucleus is called microinjection. And if the host cell is a plant cell, a direct injection is not possible. Another mechanism is used. That mechanism is called gene gun or biolistics. Gene gun or biolistics is a method suitable for plant cells, where the cells are bombarded with high velocity microparticles of gold or tungsten coated with the recombinant DNA. That is called gene gun. Then one more method is there, disarmed pathogen vector. Disarmed pathogens are also used as vectors. When they are allowed to infect the cell, they transfer the recombinant DNA into the host cell. So, all these mechanisms are referring to host transformation. Transformation means the entry of a foreign DNA into a host cell. So, the mechanisms are electroporation, heat shock, microinjection, gene gun or biolistics, disarmed pathogen vector method. Okay. The next bioprime point in this session is bioreactor. Bioreactor is a machine where the transformed host cells are cultured in a large scale. Okay. Bioreactors are used for processing large volumes of culture for obtaining the product of interest in sufficient quantities. The bioreactor provides optimum growth conditions and facilitate achieving the desired product. A stirred tank bioreactor is usually a cylindrical vessel or a vessel with a curved base to facilitate mixing of the contents. The stirring is made possible. In the sparged stirred tank reactor, sterile air bubbles are pumped or sparged. The stirrer facilitates the mixing and the oxygen availability throughout the bioreactor. So when we are describing bioreactor, a bioreactor must have 6 important components. 1. Agitator system. 2. Oxygen delivery system. 3. Form control system. 4. Temperature control system. 5. pH control system. And 6. Sampling port. These are the components essential for a bioreactor. So, bioreactor is nothing but an equipment where large volumes of cultures can be made to get the desired product in adequate amounts. And finally, once the product is produced, the product, as you know, it is called a recombinant protein. It can be cleared, preserved, and made ready for marketing by a mechanism. That mechanism is called downstream processing. The final finishing of the product by using techniques of separation, purification, and preservation, etc., before it is made ready for marketing. That is called downstream processing. In the case of drugs, certain clinical trials also need to be done before marketing. The whole procedures form what is called downstream process. So these are the prime points of this chapter, biotechnology, principles and processes. Okay, thank you.